Hello everybody, welcome back to my second video of this series, which is of the biological classifications. I'm not going to be turning my video on today because my camera is very glitchy and it's ruining the video quality. So I'll come up with a workaround. So viruses, viroids and prions. What exactly are they? They aren't a part of classification in the living world. They stand between the world of the living and the non-living, the real undead. So what viruses are like, they're between the line. So there's the living world. So you have your cow, your dogs, your humans, your plants, your bacteria, everything. So that comes on one side, and then your non-living stuff comes. That's non-living stuff is what? Paper, your electronics, everything else. So on that gradient, your virus comes between them. He has 1 foot in the living world and 1 foot in the non-living world. They are simple nucleoproteins. So viruses are basically proteins, and they have genetic material inside that protein. They have no cell organelles. They have zero cell organelles. The only thing that is there inside a virus is only a capsid and the genetic material. This doesn't make them prokaryotic. Prokaryotic comes on another side now. Prokaryotic is full for living cells, but here virus is already classified as half living, right? And they're obligate parasites. So we will come across these words which are basically obligate parasites, and you have your obligate parasites, you have your uh, facultative parasites. So what exactly is an obligate parasite? It's an organism that can only continue living inside a host cell. So the Hindi word for obligate is majboori. Okay, it's compulsory, it's mandatory. So like in Kingdom Monera, we will go through bacterial respiration, and in that you have obligate aerobes, obligate anaerobes, facultative aerobes. So what happens here is that our muscle cell is a facultative aerobe. Okay, compulsorily it has to respire aerobically, but from time to time it can respire anaerobically. So when you're running and your muscles go sore, that's them respiring anaerobically. Humans are obligate aerobes. Okay, humans cannot live without oxygen. Any animal can't live without oxygen. So here they are obligate parasites. It's an organism that can only continue living inside the host cell. Living, the viruses are living when it's inside the body of a host organism. So when it's inside the host, it's a living thing, and when it's outside the host, it's a non-living thing. And the capsid is the outer protein layer they have. So here are two pictures I have taken from the internet. Picture credits go to Encyclopedia Britannica. The first one is a strict human infecting viral cell. Well, the second one is a strict bacteria infecting viral cell. So first we'll go through the influenza virus, which is this. So there are a few shapes and sizes that one has to go through for bacteriums. So this is known as a spherical capsule. What a spherical capsid is, that it's like a 3D shape, like a sphere, and it has the genetic material inside it. And then you have your bacteriophage, which is a very, very unique shape for a virus. So he looks like a spider, like a he looks like a machine, he looks like a drone. But it's actually a virus and it infects only bacteria. So this is a universal rule. Let me write it down. Viruses are smaller than bacteria. Viruses are smaller than bacteria. This is like a very big conception that you have to follow. They're always smaller than bacteria. So here is your influenza virus and here is your bacteriophage virus. This will only infect bacteria. So what this has is double-stranded DNA. An influenza virus has single-stranded RNA. Viruses and their hosts. So when a virus is a plant host, single-stranded RNA is the only infectious nucleic material that it has, okay? Single-stranded RNA is the only infectious material it has. And it causes several diseases in the plants. So there are four examples which are mosaic formation. So what exactly is mosaic formation? Let me use green for plants. So what mosaic formation is, give me a second, line width. So you have your plant here, you have your plant leaf here, and what a mosaic formation does is it causes spots on the leaf. It causes spots like these, and they're very pattern based. They have a very distinctive pattern here. So this is called mosaic formation. So this is mosaic. Second, you have leaf rolling. So normally leaves grow this way, right? They grow normally. Thoda sa they have, little issue happens while shaping, but that's naturally occurring. What happened here for leaf rolling is that this regular type leaf, it will start growing this way. This is a very big issue because this causes problems in transport of sugar, transport of glucose. This will cause issues for photosynthesis because stomata are open, they're on the upside now, and the sunlight capturing area is below. This is the sunlight capturing area of the plant, so that is getting shadowed by this overlapping region. So when the sun is here, no sunlight is being collected by the plant. So photosynthesis decreases by a very big rate. Yellowing is the leaf, and then what happens to it is that little yellow patches start appearing on the plant. This is orange, but then little yellow patches will start appearing on the plant. And vein clearing is what it sounds like. Vein clearing is just the simple leaf, and then it loses all its veins. So the veins start clearing, the veins start thinning down, they start breaking down. So you get something like this. So that is vein clearing. And in animals, the genetic material of viruses always infect nucleus. So we will go ahead and we will find out what type of genetic material. So here you have single-stranded RNA only. Okay, plants are only infected by single-stranded RNA. So several diseases in humans, you have AIDS and herpes, rubella and mumps. So AIDS and herpes are STDs, and rubella and mumps are normal viruses. So these are your viruses and their hosts. Next, we have discovery of the viral cell, and it's done by DJ Evanovsky. Okay, this is the person who discovered the virus cell. And he discovered what virus? He discovered tobacco mosaic virus. Okay, tobacco mosaic virus. This is a virus which naturally occurs in the plants. It's a plants virus. Okay, so what is tobacco mosaic virus and how was it discovered? So we know what the tobacco mosaic virus is. Let me change the color. So here you have your simple leaf and it's infected by the tobacco mosaic virus now. So little patches will start appearing on it. So what happened by this DJ Ivanovsky is that he noticed these, deformations in the leaf. So what he does was, this is a very long process. So you need to remember the term by turn, what happened. So what he did was, first he took the leaf, he took a beaker and he created sap from the leaf. So how do you collect sap? You crush it, you grind it into a fine powder, and leaves have water in them. Leaves, obviously, all living things on this earth are kind of water based. So he collected plant sap. Okay, so that's basically a liquid leaf. So once he collected that, what he did was he took another beaker and he took something called as a bacteria proof filter. Okay, this is a bacteria proof filter. Let me label it. This is your bacteria proof filter. So what happens in the bacteria proof filter is that it has holes. Okay, every filter has holes like this. So these holes are smaller than a bacteria. They're smaller than a bacteria also. The holes on the filter are smaller than the bacteria. So later what happened is he collected the sap. So this is completely bacteria proof now. Okay. After this, he collected all this sap in a dropper. So you take a dropper, you collect all this sap. So he transferred all of it into a dropper. So you get your sap here in the dropper. And what he did was he took a healthy leaf, a normal leaf, and he put the drops of the, the liquid leaf on the drop. So what happened, what he observed on this leaf was when it was healthy, after a few, after some time, he found that there were spots appearing on the mosaic spots are appearing on the leaf again. So what did he conclude from here? He did this experiment and then another man called Beijenick, M. Vigenic. So what that man did is he concluded that this is actually a substance that is causing this issue. So he concluded from this whole experiment is that this is a substance that is causing the issue, like a toxin that is causing an issue. But then the same person, so what he did is he turned this whole process into a cycle. So he kept doing this, he kept doing this. So after doing this continuously, what he found out was now it is clear, we have done chemistry, that when you have, for example, 20 milliliters of some solution and you keep using it over and over again, and that you keep filtering it and using it, you know that the toxin is going through the leaves veins and it's going everywhere around. So it's obviously noticeable that what he's doing is the toxin concentration is decreasing. So the effect, the intensity of the effect shouldn't be this strong. So then he again concluded this substance is not causing this. Okay, so the substance is not causing the issue. Then what he said is a very famous line. Okay, it's contagium vivum fluidium. So what that says is it's basically loosely translates to infectious living fluid. And later, how was this virus formed? Was that a person called W. Stanley? This name is easy. W. Stanley, he crystallized it. He crystallized the sap and he found the virus inside it. He crystallized the sap and he found that the virus was inside this. So this is how the virus was discovered. So contagium vivum fluidum is the way he found out that this virus was present. Next, so we have genetic material in viruses. So first we'll go with the easiest one, plant viruses. We do not take plants, we take plants for granted. We don't respect them enough. So they also get some disrespected form of RNA. RNA is a very small DNA. It's half of a DNA. It's DNA cut into half. And it's also single stranded. So it's single stranded RNA viruses will cause diseases in the plants. Then you go on the other extreme. We take bacteria very seriously. We are scared of bacteria. So they get double stranded DNA viruses. So here you have single stranded RNA. So this is like, this is like a gradient. So you at one extreme pay, you have single stranded RNA. This is the simplest form, and then the most complex form, you have double stranded DNA. And in the middle, you have animal viruses. So they're single stranded RNA, they're single stranded DNA, and they're double stranded DNA. So animals get everything here. So viruses are caused more in animals here. So next you have something called as the Lytic cycle. So what is the Lytic cycle? Okay, so the Lytic cycle. So you have a simple bacterium here. You have his genetic material here. Okay. So this is the bacterium's genetic material. And then what happens is that it gets infected by a bacteriophage. Remember how I said that bacteriophages are the ones which infect the bacteriums? So it gets attacked by a bacteriophage. And next, what happens is this bacteriophage, which is there, he will transfer his viral genetic material. So, like we just did, when bacteria are attacked by viruses, they have double-stranded DNA. So, this is double-stranded DNA, as you can see. And viruses cause disease by genetic material. So, once this double-stranded DNA enters the bacterium, next what happens is that the viral DNA it destroys this natural loop of DNA which the bacterium had. So this loop of DNA is broken down into pieces. So the viral DNA, what it does is it breaks down this loop into pieces. Then that's the only DNA that's left in the cell. So after this, what happens is the protein synthesizing enzymes inside the bacteria. These are protein synthesis enzymes. Protein synthesis enzyme. So these are your protein synthesis enzymes. After this, what happens is that these DNA and these protein synthesis enzymes they bind together and they start duplicating the DNA. Okay. So this DNAs are proteins. So these enzymes, what they do is they start duplicating the DNA. So you get a lot of viral DNA in here. After this, what happens is this viral DNA will mature into baby bacteriophages. So scientifically, they're bacteriophages. So you get baby bacteriophages here inside the virus. So you get baby bacteriophage here. And this is inside the whole cell, bacterial cell. And what these do is, this is a very simple process. So what they do is they tear out this whole covering, bacterial covering. So what they do is they tear this bacteria out. So this bacteria is dead here now. Okay, the bacterial cell, he is dead. Bacteria dies here. So the once the bacteria dies, this whole process is repeated. So what these bacteriophages do is that they repeat the whole cycle. So this is known as a lytic cycle. So whenever this bacteria around and the bacteriophage attack this bacterium, this whole cycle will keep repeating. So what is this whole thing called? It's called your lytic cycle. And this is the reason why humans aren't, day and night they aren't attacked by bacteria. This is the way that God, he equalizes out bacteriums also. He is giving the bacteriums also good teaching not to infect humans and other animals much. So cholera bacterium, you have many bacteriums. This is not cholera. This is another bacterium. This is bacillus bacterium. Bacillus is a shape. It's not the name of some bacterium. This is bacillus shape bacterium. This is bacillus shaped bacterium. So bacillus is basically rod shaped bacterium. So I've zoomed into this so much that it looks like a rectangle. Okay. These corners they become square, rectangular. They become what shape do they become? Helical. So here, this is your lytic cycle. But as nature works, everyone has their own chance. We have survived for a really long time because of doctors, because of medical professionals that help us survive in this cruel world where there's viruses and there's bacteriums and there's dirty parasitic worms, there's ascaris, there's ascalmintes, there's platyhelminthes, there's very dirty organisms everywhere, and they're all coming to live inside you. So this bacterium, bacteriophage is a very dangerous parasite. So restriction endonuclease enzyme is a very helpful enzyme which helps cut the viral genetic material. It devoids the genetic material, makes it useless. So that is one way that it can be saved. The viral genetic material is, it's eliminated by restriction endonuclease enzyme. That is the first way. And the second way is the lysogenic cycle commencing. So we will do what lysogenic cycle is in the next video. I have my socials below. You can go through each of them properly. There's Instagram and there's Twitter. There's not much. I have just started making videos. So this is part one of viruses, viroids, and prions. So in the next video, we'll do lysogenic cycle, viroids, and prions. Thank you so much for watching this video. Please give it a like and hit subscribe.